Hey, this is Joel Duff, and I'm back to talk about one of the most important questions facing young Earth creationists today: how to distinguish one biblical kind from another kind. And I'm going to talk about one person's proposal that I'm going to call the divine genetic guardrails. And it involves the question of how to identify the uncrossable boundary between biblical kinds. So we've got a little genetics and taxonomy talk coming up. Young Earth creationists are constantly faced with this question as they observe the biological diversity of this world. Do any two organisms you're looking at, do they share a common ancestor, or are they separate creations? Let's take a look at an example. Here's an example from the canine group, all right, the family, which includes dogs, wolves, jackals, coyotes, and foxes. I think we'd all recognize that these organisms share similar characteristics, and that's why they're classified in a single family of organisms. That classification suggests that they all may share a common ancestor. That would be one explanation for them sharing similar traits, right? That they shared a common ancestor in the past and then have diverged from that common ancestor, but still share many traits in common. The young Earth creationist needs to ask themselves, are each one of these different species, actually dogs aren't species, they're actually considered part of the wolves. But are each one of these different organisms separate creations, or are they actually bound by a common ancestor and therefore all part of one kind? So, for example, I think that everyone agrees that dogs and wolves are actually the same kind of organism, meaning they have a common ancestor. Dogs derive from wolf ancestry. But then the question may become a little more difficult. What about jackals and coyotes? Are they related to wolves and dogs by a common ancestor? I mean, if you go back in time, there was a time in which there was a animal or population of animals for which all of these different species have been derived. And what about foxes, which are a little bit more different? Are they also part of the same kind? What are the boundaries of the kind? And we could illustrate that by asking a further question. What about all of these? Are these all the same kind of organism? Were they created with a common ancestor, then diverged into bears and hyenas and canines and felines? Or are the felines different enough from the other carnivore type organisms to be placed in a separate kind, meaning all the felines may share a common ancestor in the past, and bears share a common ancestor in the past, but they themselves don't share common ancestry, even though they share many similar traits. How much difference do organisms have to have in order to claim or identify them as being separate original creations 6,000 years ago during the 6 days of creation? That is the question that young Earth creationists are grappling with all the time, trying to find traits and features and methods to interpret biological diversity within the context of there has to be discrete kinds because Noah brought kinds on the ark, right? And therefore that suggests that they are different identifiable things. And yet the biblical text doesn't tell us exactly what a kind is or identify specific kinds. It talks about Adam, right, naming the kinds, but we don't know the kinds that he named. And so the exercise of creationists is to identify the things that Adam saw and called a kind. And then what are the descendants of those kinds and how much change have they been allowed to experience since the time of creation? Now, I think this particular slide kind of sums up the position of most young Earth creationists and puts sort of the framework on the limits of what must be considered when you're considering what a biblical kind is. This comes from a Facebook Live presentation in 2019 by Karina Altman at the Creation Museum. And she was talking about animal kinds on this earth. And so here are I guess you could say these are the main points, main things that all creationists need to or accept. They accept that life came from life, right? God created the different kinds, and since then, all kinds emanate from reproduction of those original kinds. Original animal kinds were created 6,000 years ago. That's part and parcel of the young earth creationist interpretation of Genesis, that the world is no more than 6,000 years old. So therefore, all the living things on earth can't be more than 6,000 years old. Changes within animal kinds are directed. Now that's a loaded and difficult part of this theory to explain, but it suggests God's guidance of changes over time from the original kinds to the expression that we see today. What is the mechanism of that is a huge question for creationists that are rarely grappled with. No new animal kinds have evolved. Again, that's a significant feature of this model. There are the kinds that were produced in the 6 days, and God ended his creation at that point. Therefore, no new kinds can have evolved. So any variations you see within a kind are not making new kinds. They're simply diversifications or modifications within a kind. That raises the question, how diverse can a kind become over time, right? How much can they diverge from a common ancestor? Can they diverge to the point where it becomes hard to recognize them as original kinds and they start to look like they're separate kinds? They say, no, that's not actually possible. But what are the guardrails? What prevents an organism, right? What prevents individuals or populations or one species of a kind from changing too much to become what appears to be a different kind? What are those guardrails? And we're going to discuss that in my discussion of the divine guardrails set upon organisms that prevent them from changing too much and yet allowing them to change. And lastly, modifications occur in structures that already exist. Very crucial point here. So young earth creationists recognize that organisms can change over time. And not only can they change, can a species change, but a species can diverge into two separate species, each of which have different features that are adapted to different environments. Therefore, modifications happen, right? Change happens. Evolution, in the sense of change over time, does happen, again, within these divinely appointed guardrails. How much change can happen? Well, they're going to put something of a limit on it by saying modifications can only occur to structures that already exist. That suggests the idea that an organism like a canine, right? They can modify the things that they have, ultimately modifications of their genomes, but they can't actually come up with an entire new structure, right? They're not going to come up with a different kind of lung so they can swim or live underwater or something like that. They're going to play around with the genetic information, and that's where we're going to get to, is we're going to talk a lot about information. They're going to play around with the information they have been given, and they can modify that information, but they can never have gained any new significant information. And this is the hard part of this talk. We're going to, what does it mean? New information. What constitutes new information versus just playing around with the information you have? What limits how much you can play around with your genetic information? What limits the extent of modification that you can do? Right. So these are statements that are found throughout the young earth creationist literature. They're summed up nicely in this particular slide. But the devil's in the details, right? Getting down to what exactly does that mean? What does it mean that changes are directed? What does it mean that modifications can occur, but only to things that have existed before? So let's get to the main point that we're going to address in this conversation. We're going to talk about genetic information and the origination of new information. So creationists frequently claim that new information, difficult to define word, cannot be gained by an organism. So here's a couple quotes. Duplications are the result of duplicating existing genetic information, and mutations alter existing genetic information, whether original or duplicated. Neither of them adds new information. Now that gives us a clue as to what young Earth creationists believe information is. All right, somehow information is the pre-existing, we'll call it genetic code. If you copy some of that code, so now you have two copies of that code, they're saying that's not new information because you're just copying something that was already there. And so you're not actually creating novel or brand new information. If you modify that existing genetic information, in other words, if you have a mutation that changes an A to a T, which then might change the amino acid in that protein, which may change its function somewhat, that's not actually new information, even if it happens to produce a new characteristic, because you're just modifying existing genetic information. Interesting. But let's go on before we address this in a little more detail. Evolution simply lacks the mechanism required to create new information. And I think what they're suggesting here is evolutionary theory or evolutionary mechanisms. Evolutionary theory doesn't have a mechanism at its disposal to create new information. Now, of course, we're back to the same old question. What exactly is new information? Because in the quote above, we saw that duplications aren't new information. Modifying a gene through mutations is not new information. So obviously this must be like de novo, brand new, like boom, all of a sudden there's a new strip of DNA that has a new function that didn't exist before, and it didn't come from modifying something that was there before. It didn't come from copying something that was there before. It's brand new. So it's a very restrictive definition of what new information constitutes. And I think that they're going to paint themselves into a box, as you'll see as we move along here, that, sometimes you when you try to explain something in detail, you get to the point where it starts to actually mean nothing. And I think that's what's going to happen here. For neo-Darwinism to be demonstrated, brand new specified information. So here Brian Thomas from ICRs is trying to add a more specificity, right, to the definition of information. Brand new specified information needs to appear integrated into the original and producing a novel functional structure. So a brand new function that comes from a brand new gene that doesn't have its origin in the original material is somehow now new information. This has not been demonstrated. And the connotation here is if that hasn't been demonstrated, and evolution, the change of organisms from one kind to another, involves adding new genes, new structures, new functions, novel functions, and this hasn't been demonstrated, then evolution explaining all organisms from common ancestry can't possibly be true. Now, I've tackled that last question by Brian Thomas, or last statement by Brian Thomas, that no new novel information can be integrated into an organism. I did an entire 2-hour talk called Can New Genetic Information Be Gained by an Organism? So you can go look up that YouTube video and I'll link it below. In there, I demonstrate that there are organisms that have brand new novel genes that have been obtained that are different than the original, I guess you could say, kind of organism that it was, and add a novel feature, a novel function to that organism. Won't get into that here, but I just want to state that most of these statements are inaccurate when you start looking at the details. But we're going to move on, and we're going to look at a paper very closely that's recently published that deals with this problem of identifying the discontinuity between kinds. And I think it's going to get us right to a crucial idea about genetic information that I haven't seen expressed in this way before, but I think is helpful. So in this paper, and I'm going to pull out a quote here before we go back to and look at the main thing. And so I'm going to give you the big picture summary here. There is a quote midway through the paper: "The guiding principle is that natural processes do not generate new genetic information de novo." So it talks a lot about how evolutionary mechanisms cannot make new information. And so the guiding principle behind my theory or my proposal as to how to identify different kinds, one kind from another, involves a guiding principle. And that guiding principle is new information can't be formed. All right, so that forms the foundational assumption of his idea. The absence within a group of taxonomically restricted genes, he calls TRGs, in a similar species would determine that it does not belong in the group under consideration. So what his goal is in this paper is to identify ways, what are some evidences, what are some data we can collect on different species that will help us identify these species belong in one kind, but this other species is actually a different created kind, and they don't belong together. It would be wrong to actually lump them together because they don't share a common ancestor. So here's my little figure down here. So he's saying, we got species 1, 2, 3, and 4, and you want to know, hey, are they all the same kind, or are they different kinds? I like to think of it like, let's say this is a fox, all right? And these are other canines. So we have a wolf, a dog, and an African wild dog. And you were to look at those and you say, they have a lot of similar features. I'm tempted to sort of put them all together. And if I put them all together in the same kind, what I'm implying is they share a common ancestor. So on Noah's Ark, there was only 2 of this kind that then diverged through various mechanisms, actually natural mechanisms, but somehow guided, according to Answers in Genesis, into these different species, 1, 2, 3, and 4, which now are distinct from one another and yet share a common ancestor. However, if I look in their genomes, and this is Wilson's proposal, he said, we can use a genetic criteria. If we look at their genomes, which now we have genome information, and we see that this organism has genes A, B, C, D. But we look at all of these 3 different species, and we see they have genes A, B, C, D, and E. So E is not found, right? There's no E over here. And yet E is over here. That suggests that E is a novel gene that has a novel function in species 2, 3, and 4. And since 2, 3, and 4 have this novel gene, and novelty can't evolve, right? It has to have been found. Any information, again, this word information is critical, any information that is in an organism that's in the same kind, right, has to have been there at the original creation. God has not added any new information to organisms since then because his creation was finished, all right, at the end of the 6th day. And so therefore, no new novel information can be found. And if you deem E to be novel and unique and not found in a fox, then these 3 must be one kind, sharing a common ancestor, but they cannot share a common ancestor with foxes because they don't have this novel gene E. Let's look at a more obvious example. If you were to say species 1 is felines, all right, cats, and then over here, 2, 3, and 4 are dogs, you might not be surprised to find out that there are novel genes found in dogs, all right, or canines, and there are other genes, so maybe say there's gene F over here in felines. And maybe it's gene F and gene E that are create distinctive traits that make it easy for us to identify a cat versus a dog. Right? And all the canines share this trait, but they might have modified it to express it in slightly different ways, and therefore we get different variations of dogs. And cats maybe are expressing different versions of F to give us various versions of cats. And Wilson will say, hey, all right, so there you have different taxonomically restricted genes. They could not have evolved from each other, at least that's his assumption. Or if cats have, as I was saying before, they have G, H, and I, right? Those are novel genes to cats. Well, that's something you can look at and say, oh, that's a different group. That must be created separately because new information can't evolve. All right, but can it? Right, we're going to get to that. But before I do that, I'm going to read Wilson's paper because I think it is a wonderful introduction to setting the stage for what creationists are thinking right now about how to identify kinds and what this new information thing is. So I have to stop, give you my warning. Coming up, super geeky, molecular biology, and taxonomy stuff. All right, we're going to dig in a little bit deeper. So we're going to dig way down into a young earth creationist academic journal called the Journal of Creation, Theology, and Science Series B, Life Sciences. And unless you're really into young earth creationist literature, you probably have never heard of this journal. You may have heard of the various so-called peer-reviewed journals of young earth creationist ministries or organizations such as Answers in Genesis, which has the Answers Research Journal. And yes, there are, academic science, creation science academic articles that are published there that ask these types of questions. But if you want to go look at the people that are sort of the academics behind the scenes in young earth creation science that are really thinking through these thorny questions, it's small journals like this, which maybe 100 people have read, that really explore some of these more interesting questions. These aren't articles that the average person in a church pew is going to ever have read or heard of, and they're not meant really to be read by the young earth creationist who doesn't really know what the big questions in creationism are. What it is it's meant to be read by other, I'll say, people who are in young earth creationism, who are researching those ideas, who are influential in the young earth creationist community. They're the ones that are going to know about sort of these ultra academic articles. And so these are the people that are influencing other creationists who eventually will be promoting ideas to the general public. And so this is where the back and forth, comes into play. This is where you sort of see the nitty gritty of like the real questions that young earth creationists have and that they're really grappling with. And you can see just from the titles from this paper. I mean, so this is Todd Wood. This is actually from November 2011. And this particular volume is called Creationist Perspectives on the Species. Sanders writes about minimal discontinuity species concept, a practicing taxonomist attempt to understand species in the context of baraminology. All right, catchy title there. But essentially, these are all different attempts to ask. These are different proposals for how to identify species and ultimately kinds within the creationist context. And they have common features to them, but they also disagree about exactly how kinds can be identified and how species can be identified. There's even disagreements about whether species are really an important thing to even talk about as real things. And that comes out in Wise's paper, Species as Brushstrokes, really fascinating article, the revelatory species in creation. So he has his own name for identifying species called the revelatory species. But the paper we're going to look at is Genomic Equivalence and Speciation, proposing a new criteria for baraminology by Gordon Wilson. So this is the paper. And Gordon Wilson is a professor of biology at New St. Andrews College in Moscow, Idaho. And we're going to read through some of this paper, and I'm going to make comments on it. Hitting the abstract here. Here we go with our theme. This is our major theme. This paper explores the inability of microevolutionary mechanisms to generate novel genetic information. So, right, first line of the abstract. There's the inability of microevolutionary mechanisms, that's natural selection, genetic drift, mutations, gene flow, to generate novel genetic information. Right? So if this is true, that microevolutionary mechanisms can't generate novel genetic information, he's saying that everything else that he's going to say falls from that, comes from that particular assumption, right? This has practical ramifications on how baramins are delineated. In light of the limits of microevolutionary mechanisms, all right, so he's assuming microevolutionary mechanisms have these limits, and again, this is what I'm calling the divine genetic guardrails, that prevent kinds from exploring space too far, but also provide boundaries between one kind and another kind, not allowing them to have common ancestry either. All right, it's the evidence you can see that common ancestors don't exist. All right, so I'm going to pull out a quote from the paper before I come back, and we'll be a little more methodical about it. Because the Hebrew word means kind, I might say is interpreted or translated as the word kind, we have quite a bit of interpretive wiggle room. Right? And this is notable, right? And any Hebrew scholar, anyone who's written to try to talk about what the word means is probably a frustrated academic, because the word mean is used in multiple different contexts, and sometimes it's referring to, and it's talking about a very specific organism. So it seems like a species, what we might think of as a species. And then other times it's very, very broad and general, sort of almost like birds, right? And so what exactly is a kind, if you wanted to define it scientifically? Like what is being referred to here? Are they natural groupings of some sort? And Christians say, well, yes, they have to be some kind of natural grouping, something that God made, because you have, for example, I already mentioned, Noah was to bring 2 of every kind, every mean, on board the ark, right? So that means there was 2 of some group of things out there that represented a kind, and those kinds therefore must be distinct from one another. Since there were multiple different things brought on the ark, they represent multiple different kinds. And so the task of young earth creationism in this case is to identify exactly what are the limits of those kinds and what were those kinds. And this has been a really difficult task, because the scriptures themselves doesn't provide a specific guide as to exactly what Adam named, right? So Adam's tasked, name all the different animals, right? Name the different kinds of animals. And so we'd say he named all the animals, but he didn't write those down. Like, here, this is this kind of animal, and this is the limits of that animal, right? It's like, so back to this quote, interpretive wiggle room. Yes, lots of wiggle room. When deciding how many similar, and I like this analogy, Russian dolls, all right, species, can fit into the big Russian doll, the kind. All right, so there's multiple species that may be contained within a single kind. You can think of those as Russian nesting dolls, and I think this is a great analogy. But regardless of the number of smaller dolls that can fit into the big Russian doll, we know that new genetic information will not arise naturalistically as we unpack the dolls. Let's think about this for a moment. So we have 2 scenarios here. He's saying, look, the original doll, right, the original created kind, contained inside of it, right, the genetic information that it had in its creation had the capacity to be opened up and expressed as a bunch of internally nested Russian dolls, right, or internally nested species. And the species you can kind of think of as subsets of the original whole. Right, so as natural selection and these different forces shape the original kind and separate it into different species, it's kind of like parsing the information that was in the original kind and distributing it into separate packages, which we would call species. And then, so what are the limits of a kind? How far can they go? Well, it would be limited by how much information was contained in the original Russian doll. Right? How much information was in that original genome of the original kind, and how much it's allowed to be modified. Now here's the rub. It can be modified, as we've already seen, and we're going to continue to see. Mutations can change it somewhat. There can be fairly drastic modifications of the original information. But he's saying, but we know that you can't have new genetic information. So all you can do is sort of like separate stuff out and create these nested dolls, these nested species within the kind. But now, it is what, and his question is going to be for other creationists is just how far do we extend the kind? If I see, the original kind, and then you pull out all these nested dolls from inside of it, and they're really different from one another. Can that really happen? Like, can you really get that much variation from the original kind? Or if we explore, these very carefully and we look in their genomes, we might find out that they're different enough that they weren't actually inside that original species. They weren't in the original kind. They were actually a separate kind. So maybe this one's nested in this one, but this one isn't nested in this one. And so we need to separate these out. And maybe these two are also separated out. But it's also possible that God gave this original kind an immense amount of genetic information and the tools to modify it, right? And so maybe more modification was allowed for some kinds, whereas maybe other kinds, hey, there's a certain amount of information, and when you reproduce, you make slight modifications of that in terms of adapting to the environment, but it's very easy to identify, hey, these are all members of the same kind. They all came from the original doll, okay? So what we're talking about is how to identify when you're looking at species that exist today, because we're not actually looking at the original doll put together. We're trying to infer backwards, right? That's what creationists are doing. They're trying to infer backwards from what we see in the present and try to look into the past and say, oh, all these things we see out here, these 20 different species we see, we think they're all nested together and we can lump them together into the same kind. What Wilson's going to do is say, here's my criteria. If there are unique genes and unique characteristics that we find when we look in the genome, then those unique things can't have come from the original kind, which didn't have that information. And since we know, that's the assumption, naturalistic mechanisms can't create new genetic information, whenever we find new genetic information, it has to be a unique kind. That had to have been original to the kind. And so therefore we need to split this set of what some creationists, and he is claiming this, he's saying that many creationists have gone too far. They've looked at, I got to get rid of all this ink. They've looked at something like this, and they've thought that all that variation could fit into one kind. But in fact, there's too much variation there. That variation includes new genetic information. That can't happen. We need to split that thing into two groups. These are similar to each other. These are similar to each other. And genetically, they're genetically similar enough that we can say that it is possible for them to have modified that much within the constraints of these divine guardrails of like, here's what you can do with your genetic information. You are allowed to modify it in these ways. But if you step over this boundary, you can't step over this boundary because you can't make new genetic information. So it all comes down to identifying new genetic information, novel genetic information in one organism versus another as a way of finding the difference, the gulf between between kinds. All right, back to the paper. I love this paper because I think he's really identifying what creationists think and really setting out the challenge for them. Creationists do not lack of empirical evidence showing that mutation from natural selection and mutations in natural selection are unable to generate any novel genetic information. Now, his reference is Behe. So that's kind of, in my mind, a suspect reference, since I think Behe's wrong in his projections about genetic information and change. Besides, Behe actually does accept a form of common ancestry between most organisms. And so ultimately does believe that there is new genetic information, but he would say the new genetic information is novel, created supernaturally by God through time. And so creation, so he can still refer to Behe in this way because Behe is saying that novel genetic information requires a creator to intervene to create that information. Even researchers purporting to show that evolution of novel enzymes are misleading. So now what he's going to do is he's going to say, okay, well, I know there's some famous examples out there where people say that new information has been created, but they're actually wrong. So let's look at those. In Lenski's citrate positive mutants, it was found that E. coli already had the citrate permease gene. A mutation led to its being expressed under oxic conditions. That's true. The genome in that bacteria already contained a gene for citrate synthesis or citrate use. And what happened was, and he doesn't really point that out here, he does say that a mutation caused it to be expressed under different conditions, but he doesn't point out that actually the gene had been duplicated, and then the duplicated version then was allowed to be expressed differently than the original version of the gene. That allowed that gene to be used in oxic or under oxygen conditions. So prior to this mutation, E. coli couldn't use its gene when it was in oxygen. It only used it when it was in anoxic, non-oxygen situations. And as a result, it now has a new function, right? It functions in a different condition. But he would say that's not actually new, because that's simply taking a gene and modifying that gene. So that's not novel genetic information. The genetic information for the gene itself already existed. All you've done is modified how you're using it. And he's defining that as not novel. Right? That's the critical thing here. He's defining that as that's not novel genetic information. I would say it's a novel function, but he's not going to say that's novel information. Same claim about nyelinase, an enzyme able to metabolize nylon. So in the nyelinase example, upon close examination, it was really a gain in function, not a gain of novel genetic information. He's being very, very, he's limiting what novel genetic information is to a very exclusive class. He's saying, look, there was a gene there, right? There's a gene that already existed. That's down here, pre-existing enzyme. Rather, what was this? A mutation resulted in the modification of a pre-existing enzyme. So there was an enzyme that was encoded by genetic information. A, T, C's, and G's in the genetic code. Then there was a mutation to that particular enzyme, and that resulted in a change of the shape of the enzyme, allowing it now to metabolize nylon. Now that is a gain of a novel function, right? Because that's something the bacteria could not do before, and now it gained that function, and he admits that. That's a gain in function, right? But he's saying a gain in function is not a gain of novel genetic information. And he's already defined evolution as requiring novel genetic information to have formed. But he's saying that if you gain a function, that's not actually evolution. That's just modification of previous information. And so you can modify organisms over time. And here he's admitting they can gain functions as a result of those modifications. Don't you see this as this is actually a slippery slope to essentially accepting virtually all of evolutionary biology? What do I mean by that? Let's say that you had a bacteria. And in that bacteria, you have a single chromosome, 1 million base pairs. And you say, God made that, right? God made that bacteria. He made the genome. He made the genetic information. So that's the information that bacteria has. Now, let's say that over time, there's a mistake and that genome is duplicated. And so now it's twice as big now. And we have 2 copies of all the genes. So now the genome is twice as big. What does Georgia Purdom, what is Gordon Wilson going to say about that new genome? Is that new information? Nope, that's not new information. right? That is a duplication of information that already existed. Therefore, you're not actually creating de novo information. Those genes are the same genes that already were there. So doubling your genome size isn't creating new information. It's not novel genetic information. However, if it has twice the number of genes, what can happen? You could have a mutation to one of those genes. And, oh, look right here. Here's a mutation of a previously existing enzyme that created a new function. And Gordon Wilson in this paper is saying that's not novel genetic information. He's admitting in biology, in the way the world works, this can happen, and we've proven it happens. Okay, that bacteria now has the original gene, let's say it's over here, and it has this new gene that has a mutation in it that's created a novel function. That bacteria now can do something that its ancestor couldn't do, right? This ancestor couldn't do that particular function. And now there's another mutation that occurs, another mutation occurs, and then the genome over time eventually is copied, and maybe the genome gets copied into 2 separate chromosomes. And each one of them has 2 copies of the genes, except that some of the genes have taken on new functions. But now you have other copies of the genes, and they take on new functions. And then you have mutations to control sites like the control translation and transcription. So now you regulate the genes different. You turn them on at different times or others. That's the example of the citrate, right? The citrate found by Lenski, the ability to use citrate. That's really a matter of changing when the gene is expressed. Well, that's a result of a mutation that creates a gain of function in that particular genome. So in E. coli's bacteria, in Lenski's bacteria, they gained a new function. They gained a new capacity that they didn't have before. But Purdom, but Wilson, but other creationists are saying that's not a gain of novel genetic information. But they are saying it's allowed, it can happen. Like biologically, that is possible. And where is this going to stop? Because ultimately, you can create massive genomes that have 100 million base pairs, and you can claim that all of that 100 million base pairs, no, let's say 99 million base pairs, are actually just copies of the original genome. And so that's not novel genetic information. What have you just done? You've just allowed for a bacteria to evolve into different species of bacteria, for sure. And maybe you've allowed bacteria to evolve into bacteria that clump together, or maybe one bacteria eats another bacteria, right? That has another genome that's different from itself. And when it puts it in itself, you end up with a bacteria that has its original genome, plus you have a new cell inside of there that has its own genome. And then it has some mutations, and eventually this thing becomes a mitochondrion. It starts doing the features of mitochondria. That, by the way, that's the proposed origin of mitochondria, is one bacteria basically engulfing another bacteria and then it taking its genome and using it for other purposes. None of this, according to young earth creationists, would be novel, right? Because it's all using pre-existing information in their definition of pre-existing information. And so a bacteria can evolve into a eukaryote. That is actually possible within this young earth creationist paradigm. If you just read the language that's here and you read their denial of what new genetic information is. This is they have given away the baby with the bathwater. Or is that the water with the baby? No, that's the baby with the bathwater. All they've effectively done in their original statement of saying new genetic information can't be created is they've just said life can't come from non-life. That's all they're saying. Right? At the end of the day, that's all they can actually define. They cannot actually separate one kind from another, because any kind can have come from another kind from a common ancestor without generating any novel genetic information according to their definition. All right, but let's get a little further into this. All right, let's explore this a little farther. I think we're going to see it over and over again. So how does this relate to speciation? Most creationists assume animal and plants can speciate in the short time frame, particularly during post-flood diversification, right? They're saying, oh, 2 animals can get off the ark, and they can speciate into 50 different canines, right? And so they can speciate. We know that speciation happens, and we call that diversification. Since we accept speciation within a baramin, right? But of course they're having trouble defining exactly what a baramin is. But within a baramin, whatever that is, species are allowed to form, right? Or individuals are allowed to deviate from other individuals enough that we'd actually group them into categories that we call species. You had the bigger question of like, what's to prevent them from just keeping on changing over time until you can't identify them from their original common ancestor, in which case you're going to be tempted to call them different kinds. But we must keep in mind that random mutation, natural selection, and other microevolutionary mechanisms cannot generate new genetic information in the process. He keeps saying this over and over and over again. You can't generate new genetic information, even though he's already admitted that gain of function is possible. So species within a kind can gain a new function that other members of the kind don't have. And the original kind, the original created kind, didn't have that function, right? Because if you can have a mutation to a gene that creates a new function, okay, maybe you had the original information that could be modified existed, but you didn't actually have that function. You had the potentiality for that function. But we could say that, that the original mammal had the potentiality in its genome to form all the other mammals that exist, right? Because all mammals are essentially different expressions, right, of modifications of that original mammal. And I don't see anywhere where Wilson and Purdom and others are denying that modifications creating new functions can't happen within the creationist model. Given strong empirical evidence cited above, most creationists agree on this one foundational principle. The foundational principle being new genetic information can't be generated. But as I've already pointed out, this is almost a meaningless foundational principle. The only thing it does is it keeps you from saying that non-living things could become living things. This is where we differ from naturalistic evolutionists. Therefore, this should be our major unifying premise. Honestly, that's not a difficult premise because it is so vague and in my mind, kind of meaningless, right? Because again, it just confirms something that I think is innately understood by a creationist. Life was created by God, right? The original genetic information was created by God. But it allows for any forms of modification of that information afterwards. As we follow it closely, it should resolve a number of disagreements and clear up some confusion. This proposal is really interesting, but it's not going to clear up any confusion. Based on the biological species concept, it appears that most creation biologists, including me, do not have a theoretical problem with high rates of speciation. Again, accepting this hyperevolution thing that many species are formed within just a few hundred years. As I already said, most, if not all, creationists accept branching. They accept that one species can become 2 identifiable separate species. And I think we can safely assume that this is not where the major disagreement lies. I would agree. Most creationists now completely accept that speciation happens. The disagreement lies in how we answer the following question. How much genetic information do we accept within a single barometric tree? If we acknowledge that species 1 through 10 are lumped into the same baramin due to the fact that they share A through X, then we are not thinking any differently than an evolutionist. In other words, the evolutionists are looking for shared similar things, right? Synapomorphies are called. And if there's a lot of shared characteristics, we tend to put them together and we assume they have a common ancestor because they share those things, and that's why they share them, is because they shared them from their common ancestor. And so now he's raising this idea of, hey, but there are taxonomic restricted genes. There are genes that are only found in one species, but not others. And since brand new genetic information can't form, that is our identifier. That will help us identify this is unique compared to these other individuals. Therefore, they must be in a different kind. But we've already ruined that idea of it before in this paper, because what makes it taxonomically restricted? There's a bacteria that can use has myelinase gene, but that modified from original gene in another species. Is that truly novel? He's already said it's not novel, right? So you can have taxonomically restricted genes, meaning these 5 genes, these 5 species have a gene that this other species doesn't have. Is it a different kind? Well, not necessarily, because those 5 species might have modified a gene that was in that other species, and that other species didn't modify it, and that's why it's different. Not because they were created separately and uniquely different, and that God made those genes special. Evolutionists focus on similarities because they assume continuity. That is why they ultimately concluded one evolutionary tree over 150 years ago. Creationists believe in discontinuities, but they cannot agree on where they occur and how to recognize them. Yes, he's absolutely right about that. What feature of a kind is absolutely novel to that kind and can't have formed from pre-existing information? He's going to call these taxonomic restricted genes. It's just not going to work. Disagreements over the conclusions drawn from hybridization studies, statistical baraminology, and all sorts of other methods for trying to identify different kinds. So statistical baraminology is just using lots and lots and lots of traits, mostly morphological traits, and then just saying that there are natural groupings of things that have all these traits in common, and they're different than this other group that has a bunch of traits in common, and therefore those are probably different kinds. He's saying, look, we got to get down and we actually have to identify the discrete differences between them, and they're probably going to be genetic differences. This criterion I am proposing is based on the agreed upon premise that mutations in natural selection cannot generate novel genetic information. Once again, it all comes back to, hey, we all agree you can't make new information. Like every creationist says that. New creation, novel genetic information can't be formed by any natural process. If a creationist includes within a baramin a species having a TRG that other species do not have, this implies that microevolutionary processes produced a TRG. But he's already admitted that can happen. He's already admitted that one gene can be modified to now take on a novel function, and so now it looks like a different gene that another species has. And that was by a natural process, not by special creation. This contradicts the evidence and inadvertently agrees with evolutionary assumptions. Yeah, he's inadvertently, agreeing with evolutionary assumptions. In other words, new genetic information can arise de novo. Genomic equivalence with corroboration by the synapomorphy criterion makes distinctions that I believe statistical baraminology cannot make on its own. He's actually criticizing statistical baraminology, which is a method in creationism to identify kinds. Wow. All right, that brings us back to the same quote. The guiding principle is that new genetic information can't be formed. But we're still stuck with this idea of what exactly, how would you actually identify what a novel genetic information is? How would you go about looking at a sequence in one species and then go and look at another species and you see a different sequence, or you see 3 genes that exist there that don't exist in the other species, and come to the conclusion that those 2 could not have evolved from common ancestor because it's not possible to get these 3 new genes? All right, on the surface, it sounds like a nice idea. All right, just look at the genomes. I find 10 genes that don't exist in the genome of this other species. But when you've already admitted that a genome can copy itself, modify its genes, and take on new functions, then you've already admitted that two different species will naturally have different sets of genes, even though they came from the same common ancestor. Right? You've given away evolutionary biology. You've just said evolution works. It can generate novel functions from pre-existing information. But that pre-existing information is the same pre-existing information that all living things have. Just to reiterate this point, a more extended quote from George Burr. As been said in many on this site, duplications, see, yeast failed to rise to creationist expectations. And I've talked about that article before. And mutations do not add new information to the genome. Right? I'm just saying, duplications and mutations, she's defining as not adding information to a genome. However, duplications are the result of duplicating existing genetic information, so they do happen. And so you do end up with twice as many genes after you duplicate them, if you duplicate the whole genome. And mutations alter existing genetic information, whether original or duplicated. So she's saying mutations happen and duplications happen, and all those examples where new novel functions arise are just the result of mutations and duplications of genes. So novel functions can form from natural processes, and they don't even require, in their definition, novel genetic information. Neither of them adds new information. I'm totally confused as to what this new information is. I really am. Come up with a theory. Come up with a model, a way of identifying brand new information, and how to test that. That's what young Earth creationists need to be working on. All right, so I'm going to double down on all this. So let's take a sequence. So I said, let's say in the original creation, this is what God created. God created a sequence, 4 T's, 4 A's, 4 C's, 4 G's, in a particular kind, all right, in an organism. And if you want to think of it as like just a basic bacteria, you can think of it that way. And then these are the things that are allowed to happen to that. You can take that and you can modify some of the original bases. These are individual single nucleotide polymorphisms where a base changes. The A changes to a G, A changes to a G, the C changes to a T, a G changes to an A. This potentially could change the function of what this codes for, right? This is code. You're changing the code. Now the code is going to be read and turned into potentially a protein. That protein might be different than the original code coded for. And so therefore could have a new function, or at least a modified function from the original function. Right? I don't know, no creationist is saying this can't happen. You could take the original code and you could duplicate it. Oh, I meant to, let's go down here. You could duplicate the whole thing, right? So now you have 2 copies of that code. And if you have 2 copies of the code, well, then you still have the same information you had before, but now you have 2 copies of it. Now, what if you took one of those copies and you modified it? And so this one has a new function and this one has the original function. Well, now this is by definition, you have 2 genes. One gene for one function, another gene for another function. I don't know how you can say that's not a creation of new genetic information, because you have not only a different set of sequence than you had before, you also have a different function. You have gained a function you didn't have before. That's adding to the genome. That's adding to the capacity of the organism to do things and modify themselves and change and adapt to their environment. Where's the guardrail, right? This is my original comment was like, what is the divine guardrail that prevents this from ending up making hundreds of novel new functions for an organism? And if you create 100 novel new functions for an organism, is that organism really the same as its ancestor? right? It's definitely a different species, but what if it's so different? How are you gonna not think of it as a different kind of organism? How is it not going to escape the boundaries that a kind that surround a kind? I don't know. The other thing that can happen is you could insert sequence, and so in this case, we've added CGA. Right, so we have a mutation where we add some sequence. That adds an amino acid to this protein, which again changes the function of it. And then over here, what I meant to do is I meant to actually add several new bases on the end too. All right, so what we're saying is the original created information can be modified in all these different ways. And from my reading of young Earth creationist literature, they're saying, yes, 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 you can do all these things. All these things are possible, and none of these things are creating new information. But they are modifying organisms. They are changing the functions of traits. They are creating new traits for organisms that they previously didn't have. But they're saying, this is the original doll. There's the original doll. And out of this doll, you can create these nested things. The problem with it is it's not really creating subsets. It's actually expanding the original doll into something more than it was before. And then when you say it that way, well, that's actually creating new genetic information. That's creating novelty, right? It's not a subset of the original created kinds. We're not just parsing it into separate little piles. And I think that's what the created by, the speciation process by which some creationists say, like, there's just lots of variation in the original genome. And then that variation is sorted out through natural mechanisms and put into separate little piles. And those separate little piles we call species, but they're all subsets of the original kind. But once you acknowledge you can modify the original code, not just segregate genomic variation, once you can modify it and you can create new function, it's not subsets. You're creating new things. They are evolving into something different that the original kind wasn't themselves. Now, you could say they had the capacity to become all those other things. But once you say things like that, you're really just a theistic evolutionist, right? You say, like, the genome has the capacity to change, and it has the capacity to add new functions. It has the capacity to duplicate its genome and to then modify all those duplicated genes in order to take on new challenges the environment hits them with. And over time, that's going to accumulate into new novel functions that organisms have. And then those genomes that now have all that novel, those modified genes, they then are going to be hit by new changes in the environment, and they're going to modify themselves, right? Descent with modification, right? This is just descent with modification. Now, Robert Carter and Paul Price over at Creation Ministries International, they do have an interesting article, what would count as new information in genetics? And I thought, okay, great. They've identified the problem. What exactly is new versus just reshaping, modifying something that already exists? Is there a difference between these two things? I was kind of disappointed by this article. We often start with the argument, Darwinists rarely, if ever, talk about information. They're quick to point out that DNA can change. Thus, they claim that there is no information in DNA, and the information we can see can be seen to change in a Darwinian fashion. Some creationists like to say mutations cannot create information. They only destroy information. Yes, you hear that all the time, like genetic entropy, like mutations always just break information. They never add to it. But this is a very weak argument. So here is a young Earth creationist that's admitting you're going down a little bit of a scary path here. You don't want to go this direction. All the evolutionist has to do is point out an example of DNA duplication, and suddenly there's an increase in information content. Yeah, I would say that a duplication is increasing information content, despite what Wilson and Purdom have tried to say, which is that's not actually new information. They've just defined it away. It is true that the information content of the cell can change, and it is true that mutations may add information to the genome. Great. Here's a fantastic quote showing a creationist who's saying, yes, you can add information. However, now, of course, here's the but. As I wrote, the changes we see are not the types of information gaining mutations necessary for large-scale evolutionary processes. So here's a more nuanced view. They're saying, yeah, okay, mutations can change genes and give you some new functions. Duplications can create new information that organisms can use to adapt to their environment. However, there's still a divine genetic guardrail, right? There's still a limit to how much a particular species can change. They can modify and adapt themselves within some limits, and they can become different, but a bacteria can't become a eukaryote. A fish can't become an amphibian. A dog can't become a cat. Of course, that's evolutionary biologists wouldn't say that dogs are becoming cats anyway, but you get the idea. One kind can't become another complete kind of organism. There are several known examples of mutations that allegedly cause a gain of function, but these arose from corrupted genetic information. And it goes on to talk about various forms of possible new functions. But we've already said that most creationists now would say that have actually argued that new functions can arise, but they're just modifications of old functions or old genes. What would a real, genuine increase look like? All right, to get back at the skeptic's main question. What would real increases of information look like? I'd submit to answer this question. Just sit at a computer and watch yourself type a paragraph in a word processor. Mutations are incremental. They are small changes that happen in a stepwise fashion as cells divide and generally multiply, and generations multiply. The genetic codes consist of letters A, T, C, and G, just like our English language has an alphabet. But here's the central problem. It takes hindsight to recognize whether function or meaning is really present. Watch this transformation. And then here's this example. You type H. You type H-O, that doesn't really have a meaning yet. H-O-U, that doesn't really have a meaning yet, right? It doesn't have a function. It's just information, right? And it is novel because you're adding those bases, those letters. And then you get to house. And then you say like, oh, now house has an actual meaning in the language. But all the parts before that didn't have a meaning. But now suddenly the combined effect has a meaning. At what point in the series did you understand the meaning? Perhaps you got to step 4, but you would have been lucky, for you did not know the word would be house or housing or household was about to appear. It didn't become totally clear until step 5, when the full word was spelled and the program ended. There's no real way to say before you've already reached step 5 that that genuine information is being added. When I say genuine information, so he's saying that the individual parts aren't information; they're just random pieces. So he's like, mutations just create random bits of stuff, and they don't become information until they actually are usable, right? Until they form a function. This is a little bit different view of what information is. Mutations suffer from the same problem. But there's an even bigger problem. In order to achieve a meaningful word in a stepwise fashion, let alone a sequence, sentences and paragraphs, it requires foresight. In other words, you have to know where you're getting to. And since mutations are random, they don't know where they're going. They can't really make useful information. I've already known I want to say house before I begin typing the word, but in neo-Darwinism, that isn't allowed. Okay. Right. Neo-Darwinism, we would say that an organism doesn't say, look, I need to be able to make antifreeze to survive in this cold environment. So therefore, I'm going to make these 10 mutations. And then after I make those 10 mutations, I'll have that function. So I start doing it like with foresight, like here's what I need. That's true. Mutations must be random and unguided. Due to the sheer number of possible nonsense words, you cannot expect to achieve any meaningful results from a series of random mutations. There we go. There we hit the nub of the problem. He's saying, look, you can type letters just randomly, and you're not going to make sentences, right? And so mutations are random. They don't know where they're going, and therefore new information, which is, going back to the word household, meaningful word. In order to achieve a meaningful word, you can't make a meaningful new contribution through this random process. Now, going back to, well, then how do you make a nyelinase? Oh, well, that was already an enzyme. God had already given a meaning, a purpose to a particular sequence. And because that already had meaning, it was possible to modify that meaning, which then creates a new function. But you had to have enough, like the word had to, like you have house. Now you can add hold to that and make a slightly different meaning to that word. But you can't make the original word house through random processes. In other words, you can't make a gene from nothing. But once you have a gene, you're allowed to play with that gene, right? God says, I made the genes, right? I made the original functions. And once I made these original functions, they can be adapted, right? Mutations, it doesn't require a bunch of random mutations to make the original gene. That's impossible. But you can take an original gene and through playing with it, through mutations, even random mutations, you might stumble upon new variants that actually have different meanings. So I gave you the language, all right? And then you can play with this language. So I made a kind that has this certain set of genes, this certain set of genetic information. And you can take that genetic information and now you can play with that information by copying it, shifting it around, putting it in different orders, turning one on, turning this piece of information on or off at different times. And maybe mutations even cause those changes. And you can modify your organism, and that's what allows you to adapt to your environment. None of that is different than anything about molecular evolution. I mean, it's just that is basically molecular evolution over time and how it can accumulate to make different species. I still don't see where the guardrail is, though. I don't see how you can stop that from becoming something really different, because eventually you're actually admitting you can add new genes. Because the original information, I'm going to go right back. I'm going to say it again. If you start out with 20,000 genes and you duplicate some of them and then you modify them because you can, and then they take on new functions, and you're saying all that's okay because God created the original set and you can play with those through naturalistic mechanisms, then eventually you're going to have 22,000 genes, and the organism's going to have 50 new things that it can do that its ancestor couldn't do. And then it's going to lose some of its genes. And so it loses certain functions, but now it's gained other functions. And that makes the organism a very different organism than it was before. How are you going to stop it? Like, how do you stop this process from generating novel organisms that truly seem to be unique from other organisms? I don't think you can do it in this particular model. He's created a scenario that doesn't acknowledge the complexity of the genome and how genes can evolve just from nonsense sequence, right? From random sequence. There's a lot of evidence just from the last couple of years and some very interesting articles that show how random sequence, of which there's a lot of what appears to just be random sequence in genomes. Remember, we only use a small portion of our genome to read code, to read into genes. Over just the last couple of years, we found some really intriguing evidence of genes that are actually developed from what we call the junkyard, right? That what it looks to be random sequence, and then slight modifications to that random sequence suddenly pop out new functions. This is a really fascinating summary article in Nature that I will link to, and it has to do with this particular paper, Molecular Mechanisms and History of Nonsense to Sense Evolution, taking nonsense sequences for which There was no protein made. There was no function for this particular portion of the DNA code, right? It's just a jumble of A, T, C's, and G's. And then creating a sense out of it and suddenly finding a use for it. In this case, the evolution of an antifreeze glycoprotein gene, which just appears in the genome compared to its relatives. Now, for Wilson, hey, this new gene looks like a brand new function and therefore is a TRG, and therefore those fish should be a different kind than these other ones because they don't have the capacity to do it. Problem is, they have the same portion of the genome that this other organism has. But this other organism does something with that part of the genome. It has a function for that portion of the genome. This other organism doesn't have a function for that part of the genome. So it's really just a modification of genome. It's not an absolutely novel new feature, except that it is novel to these fish. They've taken nonsense sequence and they have turned it into a sensical thing through mutations and natural selection. This isn't the place to go over all the details of how those studies work, and I've gone over it in another place at another time. How about this one? Rapid evolution of protein diversity by de novo origination in Ariza. So I love the word in the title, right? De novo, brand new origination in rice. And this is also from 2019. They sequenced the genomes of many different species of rice. And by sequencing all those genomes, they discovered that they counted up the total number of genes in those genomes. And for example, we detected, where is this down here? We detected at least 175 de novo open reading frames in the focal species Ariza sativa, subspecies japonica, which were all detected in RNA sequencing. What they're saying is there's 175 locations that are open reading frames, meaning those are frames that are read into proteins. And so we call those genes. They identified 175 potential unique genes to this particular subspecies. And they're all detected by RNA sequencing, meaning they actually make the RNAs. They transcribe them. And so probably have functions for those. And so they looked at multiple different genomes, and what they found is that among these 13 different genomes, each of them had different sets of open reading frames. So they had unique genes relative to each other, even though they're very similar species. They used ribosomal profiling, showed translational evidence, meaning they're actually making proteins from these unique open reading frames that the other species, closely related species, doesn't do. It doesn't use that particular sequence for that. 50% of them de novo genes. So most of these sites they identified as probable genes, they were able to find evidence they're actually using and making proteins. They probably have function. In recent divergence of Ariza, an average of 51.5 de novo genes per million years was generated. So they're estimating how old these species are. And they're saying to get this number of new genes, we're suggesting that there's 51 new genes formed in these lineages per million years. And so 51 genes, 51 new genes that didn't exist in the common ancestor between those two species have arisen in the time of that lineage. So let's draw this out. Species 1, species 2, common ancestor. There's 51 different new genes that this organism uses that this one doesn't. And actually that could be, what could have happened is you could have 25 new originations from this common ancestor to here, and over here you could have had like 26 new different open reading frames formed. These are considered to be mutations that occurred out in just the generic sequence, which happen to create new start codons or new promoter sites, which then turned on sequences, allowing them to use those sequences in that species, whereas the other species doesn't actually use those particular locations. I don't know what Wilson would do with that. I mean, there's that many novel genes that don't exist in another species, then he'd probably want to take those, according to his model, he'd want to put them each into different kinds. But then you'd end up with like 13 different kinds of rice that are all different species from each other, and even different varieties of rice. I don't think he's going to want to do that. But if you put them all together, then you're going to have to say that the genome is capable of turning on and creating new genes, all right, from random sequence over time. Here's another paper, Real-Time Evolution of New Genes by Innovation, Amplification, and Divergence, and this is their more typical form. There are genes, and this is something creationists I find don't recognize a lot of times. They think of one gene as having one function. There are genes that have two functions. All right, function A is the primary function. This is the main thing it's responsible for. But the same enzyme might actually have a second function. So all enzymes, actually most enzymes catalyze one primary thing, but they also may be able to do a second biochemical process. And so that function might be, we call it B, but it's not quite as important. It's the secondary function. And so it's doing two different chemical reactions. One of them, most of the time, maybe it's the most important thing for that organism. And so B can never really, it's maybe beneficial to the organism, but it can never become the primary role because it'd have to displace the primary role A. But then what happens if you copy this gene and now you have two copies of the gene? They're both doing both functions. But then if you have this gene, this figure is too complicated, unnecessarily complicated. I'll just summarize it as, if you have two copies of the gene and they're both doing both functions, what if you have a mutation in one of these that allows you to become less efficient at A and more efficient at B? Well, then B will become the primary role of this gene and A will be the primary role of this gene. In fact, once you are able to do B, you no longer need to do B in this one. You might have another mutation which then excludes B and now you only do A. So you can take the genetic information, so now we'll use the word information. This gene had the information for doing both functions. But now you've duplicated it, separated it, and now you have two separate genes, each with separate functions. And this probably creates a more efficient organism than the original organism does. And so this is how new genes, right, can form over time. Because even the dual function could be something that arose in time. It could just be, it had function A, and then there's some sloppiness to the chemical reaction, and the organism's like, hey, once in a while you do this other thing, and I can actually use that. Like in this environment, I'm stressed, and you happen to be able to metabolize this other thing to make me a product that helps me survive better, right? By chance, you now have an added function to that gene. And then you can make that function even better by duplicating it and then separating the functions. So now you do both functions really, really well. Right? That's the gradual evolution of the genome such that it gains functions or better functionality over time. And this is the paper that I spent an hour and a half talking about in my other talk, eukaryotic acquisition of a bacterial operon. So you could say, hey, all the other examples you just showed, those are examples of you already have sequence and you're just modifying it. That's not really novel. Right? You have a kind of organism and it's just modifying itself. What I want you to see, what I want you to show me is you have a kind that was created by God, and then you add something brand new to it, something it couldn't do, something it was never capable of before, and you add new DNA to that organism that has new functions. I'm willing to call that novel. Right? That's what one of those quotes I brought up in the second slide. Says that has not been shown. So then I refer you to this article, Eukaryotic Acquisition of a Bacterial Operon. This is an example of a yeast that has captured a portion of a bacterial genome into the yeast and then used that particular set of genes to derive a function from them. And so that's a new part of its genome that didn't exist before. So there are related yeasts that are very similar that don't have this chunk of bacteria in them. And that bacterial chunk is being used by that yeast, and it's part of that yeast. And now it copies it and gives it to its offspring. So you imagine this yeast is living, doing its thing, and then it gets this piece of DNA from a bacteria, incorporates it into its genome, learns how to use that portion of the genome to give itself an entire new function, which is the ability to sequester iron from its environment. That gives it a competitive advantage against its neighbors, which can't do that. And so it goes on to live, and then it speciates into many, many different species and shares that particular bacterial sequence with them. That is the gain of a new function, right? And it's completely novel to that yeast. But wait a minute, you're saying, but that was originally a sequence that came from somewhere else, so that's not actually creating new information. It is new information to that kind if you're going to define kinds as separate creations. So that shows that a kind can obtain new information. You can still kick the can back down the road, right? You can say like, yeah, but you haven't shown where that information came from. Once again, I'll tell you, then all creationists are doing is saying, okay, you can modify organisms in any which way. They can add new functions, they can subtract functions, they can gain new functions, right? But ultimately, every function has to have come from somewhere. It's a modified form of a previous function. And so somewhere the original function came from, the original ability just to live. And so that's the origin of life. And if that's what they want to do, then they can go ahead and accept Darwin's hypothesis that all organisms are related by a common ancestor, because that common ancestor is a living thing, and then every organism on Earth is just a modified version of that living thing. And Darwin didn't address the origin of life. All right? I mean, I'm sure he thought that the origin of life could be explained through naturalistic principles, but that's a separate thing, inorganic to a living thing. All right, wearing down on this, but last slide. So we're back to our original quote: the guiding principle that naturalistic processes do not generate new genetic information de novo. And I think I've hopefully shown you that's kind of a meaningless statement, because they've made this idea of de novo new genetic information so exclusive that it doesn't really help as a factor for determining what is a kind and what is the separation of kinds. The fact that one species has different genes than another, since genes can evolve from other genes, they can be lost from genomes, and they can be duplicated and added to genomes. And creationists acknowledge that all that can happen. Wilson's attempts to separate kinds based on the presence and absence of specific functions or particular genes is not really going to hold water over time. It's going to be impossible for him to identify this as special creation versus naturalistic changes to genomes over time. All right, that's an attempt to find, or I guess I'm not trying to find the guardrail that is the divine guardrail that separates kinds. I guess I'm saying I don't think that Wilson's proposal has found the guardrail. It neither has found the guardrails that distinguish one kind from another, but it also doesn't give you the guardrails that prevent species from changing into new species or kinds changing into new kinds, because there aren't any limits to how different an organism can become if it's just modifying what it has, because the possibilities of modification are nearly endless in a genome that has thousands of genes to begin with. So let's leave it there. It's still a little bit of mystery what novel genetic information would look like and how we would identify that it came into existence via supernatural creation versus providential processes that God placed in his creation. I'm still not seeing the mechanism or way to define those two things. All right, let's quit there. Thanks a lot for watching. Bye-bye.